Recall that in order for individual cells in a multicellular organism to form tissues, organs, and the organism itself, a number of things must occur. Among those are the intercellular communication that we recently spoke about, as well as cell-to-cell associations and cell to the extracellular matrix adhesion. The previous video you watched introduced the cell-to-cell and cell-to-matrix junctions, but I wanted to provide a little more information about each of those, beginning with the cell-to-cell junctions. There are three general types of cell-to-cell associations. Among these are tight junctions that essentially rivet cells together. In epithelial cells, those rivets are near the apical surface of epithelial cells. And tight junctions are maintained by associations between claudin and occludin proteins. There are two types of anchoring junctions, the second general type of cell-to-cell association. These anchoring junctions anchor cells together or link cells together by transmembrane proteins that also associate with the cytoskeleton. Finally, there are gap junctions that create channels or pores between adjacent cells such that small molecular weight water-soluble molecules can freely pass between adjacent cells, allowing for very rapid communication without involving release of communication molecules into the extracellular space. This table summarizes the types of junctions as well as the proteins involved in forming them and briefly describes the function of each type. Anchoring junctions come in a number of different flavors. Adherens junctions and desmosomes anchor adjacent cells to one another using single-pass transmembrane proteins known as cadherins. These cadherin proteins associate with intracellular adapter proteins. These adapter proteins form a dense region in the cytoplasm that is referred to as a plaque, and we'll see these plaques on subsequent slides. The plaques link the cadherin protein to various cytoskeletal elements. Anchoring junctions also associate with cytoskeletal elements via plaque or adapter proteins in cell-to-matrix associations known as hemidesmosomes and focal adhesions. Since we're focusing on cell-to-cell associations, let's talk about adherens junctions and desmosomes. One type of anchoring junction is an adherens junction. Adherens junctions depend upon the single-pass transmembrane cadherin proteins. Cadherin proteins are so named because their binding is calcium dependent. The first two letters of the word cadherin are an homage to their calcium dependence. The structure of cadherin proteins is maintained when calcium is bound. And because structure is related to function, it's only when calcium is bound that the proper structure enables binding to another cadherin protein. On the cytoplasmic side of the membrane, various adapter proteins assemble with the cadherin cytoplasmic region to create a dense structure known as a plaque. That plaque links the cadherin protein to various cytoskeletal elements. In the case of adherens junctions, the cytoskeletal element that's linked is actin. Cadherins on one cell associate with cadherins on an adjacent cell to form an adherens junction. Cadherins must be the same on each of the two cell types involved in an adherens junction. This implies that different cell types have different cadherins. The cadherins on epithelial cells are known as E-cadherins, whereas those on neuroglial cells are referred to as N-cadherins, for example. Only cells with the same type of cadherin protein can form adherens junctions. Cadherin proteins are not only associated in adherens junctions, they are also involved in the formation of desmosomes. Desmosomes link cadherin proteins via different adapter proteins to intermediate filaments. In epithelial cells, those intermediate filaments are comprised of keratin. Here is a diagram of a desmosome. The adapter proteins, as you see here, that form the plaque are different in desmosomes than they are in adherens junctions. That allows for association with different cytoskeletal elements. I don't care that you know the names of these adapter proteins, but I do care that you recognize that the adapter proteins in these two types of anchoring junctions differ. The cadherin proteins link, in the case of desmosomes, the intermediate filaments of adjacent cells by binding to other cadherin proteins, the same type of cadherin proteins, on adjacent cells. Gap junctions are created by the assembly of transmembrane connexin proteins. 6 connexin proteins assemble into a connexon, and a connexon creates half of a gap junction. You see that there are multiple types, as indicated by the different colors of green, multiple types of connexin proteins. Connexons can be created by either the same type of connexin protein to create a homomeric connexon, or different types of connexin proteins creating a heteromeric connexon. A connexon on one cell will associate with a connexon on an adjacent cell in either a homotypic or a heterotypic way. When two connexons bind to one another, a gap junction is formed. Gap junctions create a gap between adjacent cells, thus their name. Gap junctions create a channel between adjacent cells that functions in intercellular communication. Let's transition to cell-to-matrix associations. Cell-to-matrix associations occur in one of two different ways. Here we see an image of a hemidesmosome linking integrin proteins to cytoskeletal elements. In the case of a hemidesmosome, the cytoskeletal element involved are intermediate filaments. Integrin proteins are the transmembrane proteins that link via adapter proteins to the intermediate filaments. Integrins are heterodimers, always created by the association between an alpha integrin and a beta integrin. These are single-pass transmembrane proteins that associate with the extracellular matrix on the outside of the cell and with adapter proteins on the intracellular side of the cell. All functional integrins are alpha-beta heterodimers. There are many different alphas and many different betas that can associate with one another in a different way to recognize different extracellular matrix components. This table shows just a few alpha-beta heterodimeric integrins and the cognate ligands that they will associate on the extracellular side. Just like there are two types of cell-to-cell anchoring junctions, there are two types of cell-to-matrix anchoring junctions that vary based upon the cytoskeletal element that's involved. While desmosomes are associated with integrins being linked to intermediate filaments, focal adhesions are formed when integrin proteins associate via adapter proteins to actin filaments inside of the cell. The same alpha-beta heterodimeric integrins can be used to link extracellular matrix proteins to actin filaments as link integrin subunits to intermediate filament proteins. It's just that different adapter proteins are going to be used. While hemidesmosomes are involved in very tightly anchoring epithelial cells to the extracellular matrix, focal adhesions are much more transient. That has to do with the instability of the actin filaments. Focal adhesions then are involved in cellular migration. They form and they go away over and over again, just like the cell would be taking steps along the extracellular matrix. The focal adhesions are highlighted in red on this immunofluorescent micrograph. Integrins are not always very tightly bound to the extracellular matrix or to the intracellular adapter proteins. In fact, binding to the extracellular matrix enhances the affinity for cytoplasmic adapter proteins, and binding to cytoplasmic adapter proteins increases the affinity of the integrins for the extracellular matrix proteins. So in this way, a tight linkage between the adapter proteins and the extracellular matrix proteins can only occur if both things are bound simultaneously. Finally, this image summarizes most of what you've heard in this video and the previous video about cell-to-cell and cell-to-matrix adhesion. What's labeled in this image, however, as the actin-linked cell-to-matrix junction is actually referred to as a focal adhesion. The next video will introduce various components of the extracellular matrix.